and see the slides to talk about my kind of side project. And the side project is Phage Taxonomy, where I am the current chair of the Bacterial Viruses Subcommittee of the ICTV. So let's just dive straight into it. And obviously my keyboard isn't working, so I'll use my mouse to change slides. So what I wanted to talk to you today, and what prompted the invite to talk to you today, was basically this top paper that you can see here that we just recently published, which is the taxonomy update that we try to do every year, but sometimes fail because our lives are so busy, from our committee. And this has two really big changes for Phage Taxonomy, which I will present to you today. And it kind of pulls in some information of two earlier papers, the roadmap for genome-based Phage taxonomy, which was published in 2021, and the informal guide to how to name and classify your phage, which was published in 2017. So this new information kind of overrides some of this because now it's official. So let's just start with the major change. So the abolishment of morphology-based taxa. What do we mean with this? If we look at see if I can change to my laser pointer. Okay, if we look at the figure on the right here, this came from the roadmap paper from 2021. So this is a phylogenetic tree of about 2,000 phages and archaeal viruses that were put together with a program called Gravity. And there's two annotations on this. On the outer ring, there's families that are based on...
Evelien,
yes.
We are not seeing your change of slides. Have you changed the slides?
Oh, yes.
Yeah, but we're not, we're just seeing you in the introductory slide.
Okay, let's just take the laser pointer off and see. Okay. What slide are you seeing now?
Still the introductory. I
I'm
don't know
gonna
if it's the same for everyone.
I'm gonna stop sharing and
then share again.
Start and share again.
Now we can see you.
Okay. Where's my Zoom button now? I've never had this before. Okay. Can you see the new slide now?
I am seeing the taxonomy update for African Phage Forum. I don't know if it's the same for everyone. Just the introductory slide again.
Okay.
Now we can see, I can see the second.
We can see it now.
We can see it now. Now we're on the paper.
Okay.
I think it's because it's not on the projection mode.
Yeah, probably.
Okay. So is it so can you see it now in the full presentation?
No, it's not in the presentation mode.
We can, it's not in presentation mode.
I wonder if there's just a lag at the moment, because I'm in presentation mode at the moment. But let's see what.
Let me see, because I pulled multiple people sharing the screen. Maybe I'll just put one person and just say the moderator.
Okay, but then that's just you. I'm just gonna stop again. Okay. Apologies. I can see from the screen sharing button that I'm in the presentation mode, but you all just see my PowerPoint. for some reason.
Okay, let me do one thing. I'll do one participant can share, and then I go to advanced sharing options, and I'll see what. But Manuel, can I make also Evelien a co-host?
Oh yeah, that might work, and I will also just share.
So I can make you. Okay, I can make you the host. But then there'll be no, you can't, because you will have to admit people in.
You can, oh yeah, then it will share.
I think she can. We can see her slides without the presentation. You can go ahead. I think
I'm just gonna share my desktop instead of the slides, and then that should work.
All right.
So if I now do this.
Angela, if it doesn't work, make me
now it's in,
Now it's working, right?
Yeah, it seems like it's in presentation mode.
Okay, and I can go forward. Can you see the slide with the tree on it?
Abolishment of morphology based, yes.
Ooh, excellent. Okay, so if you didn't see this before, these are the papers that I was talking about. So there's three papers that are kind of the main subject of this presentation. And then, so let's go back to this tree which you can hopefully all see now. So inner ring is, in this case, the morphotype being either photovirus, cyphovirus, or myovirus. And the outer ring was the situation before 2021, when this paper was published, where we had a couple of genome-based families that we created. And then you can see that one of the things that happened, and we've been talking about this for over 10 years as a community, that with more genome sequence, we have more information about genomic diversity. And then we can see that, for example, you have these pockets of other morphotypes interspersed with, the other, so the Myoviridae, Podoviridae, Cyphiviridae families were not what we call monophyletic, so coming in one clade here. They were kind of spread across. But there are clear groups that belong together, and there are also groups that belong together where you have unrelated phages in the same family, but then related phages in different families. For example, the most commonly used example is P22-like phages and lambda-like phages, which have different morphology but are clearly related. So what we now have done is we've abolished these families, Myoviridae, Podoviridae, Cyphiviridae. They no longer exist. And so that we can really use the genomic information to generate new families. So this artificial morphology-based description is now gone, and you shouldn't use it anymore. But what you can still use is, say, give the morphotype information. Say, I have found a podovirus, which is a phage with a short tail, but that means that you don't really know yet what family it is in based on that information alone. You need the genome information. The other thing to make kind of space for more diversity is that we remove the order Caudiviralis, but it's replaced with the class Caudiviricetes. So all tailed bacterial and archaeal viruses are now in this class Caudiviricetes. So you may ask what happened to all of the phages in the old families. For some, we've been able to, at the same time, generate new families based on the genome, extensive genome analysis. So one of the latest new taxonomy proposals created two new families, Straboviridae and Cyanoviridae, for the T4-like phages, where the original T4-like phages are in the family Straboviridae, and those cyanophages that are T4-like are now in the family Cyanoviridae. So if you're interested in, the type of analyses that come with this, creating these two new families, which have a lot of members, and you can see that for each, leaf here being another genome. You can look at taxonomy proposal 2021-82B, which is available from our website. And one of the things that was done for this, to create this tree, is based on the phylogeny of these core genes that were identified as being orthologous proteins that are shared across all of these phages, and that can be used to build a phylogenetic tree, which is more of a phylogenomic tree in this case, because you use more of the genome. But these are now only 2 families, but what about all of the other bacteria phages? So all tailed phages are now associated or assigned to the class Caudiphyracetes. If we don't know anything other about genus, subfamily, family, we'll just say this is unclassified Caudiphyracetes. There's now many genera that were created that were in, for example, the family Myoviridae, family Cyphiviridae, and we haven't been able to provide to get new family information yet. They are now directly assigned to the class Caudiphyracetes. And then we are gradually establishing new families if we can establish clear demarcation criteria. What do I mean with that? With clear demarcation criteria, if we can say that they're clearly distinct from other families, and that we can make clear criteria on that other people can use to evaluate the inclusion or exclusion of new phage genomes or new phages in these families. So what we want is to have enough members to define a core genome, to define which genes are core, so which genes are shared across all of the members, or a clear set of features that separate the family from others. So we have in last year, there was a family called Naumviridae that was created with only 2 members, but these 2 members had so many features that were distinct from others that it was clear that we could create a new family for just two phages. But practically, the most new families, they will need at least 2 genera with each have multiple isolates and species to make it clear what the criteria are. So what's the current situation? And somebody drew on my screen, but I don't know if you all can see that.
I think I want to pull someone out. We might have the same situation we had last time. Okay.
So what's the current situation now? This figure is also in the paper for our taxonomy update, and this shows us the realm, kingdom, phylum of the class Caudiphyracetes, and this is not a phylogenetic tree. This is just a dendrogram to indicate where everything groups together. So in this class Caudiphyracetes, we have in blue some archaeal virus families, and in green the new genome-based families, and they incorporate all of the taxa that used to be in Myoviridae, Podoviridae, Cyphiviridae. So there used to be 625 species in Myoviridae, 130 in Podoviridae, and 1166 in Cyphiviridae. These have now been distributed among the undefined and the other families that have been created. For example, in the previous year, the family Schitoviridae was created for phages that were related to N4. So we've now added 8 genera and 9 species to this family in this update, and they came from in here. So here you get an idea of which groups have the most phages in them, and that's currently the undefined group. But that's just the way it is, and we'll slowly start chipping away at making them less undefined. This is another way of showing this kind of chaos, or the order to the chaos that is currently being made. So this is now a Viptree tree that as you can download it or see it on the Viptree server online, which has 4,000 plus double-stranded DNA phages. And what I've done is I used the inferred database to annotate in the inner ring the new families and in the outer ring genera. And then you can see that there's actually unclassified is in gray. So certain areas of the tree, like this one, we've done a pretty good job in annotating this. And this is those Straboviridae and Cyanoviridae here. So we've done a good job. This area of the tree, not so much, because there's a lot of gray here. So we still have a lot of work to be done. But for example, this is the genus Chaoctovirus. So looking at a lot of phages that belong together here. So maybe it might be not too difficult to create a family for these. So this is the kind of stuff that we're working on right now. So you might have some practical questions for any of your own phages that you have. So if you have a new phage genome, or new phage, you've sequenced the genome, you've done a high quality assembly and annotation, then you can start asking, where does it fit into the current taxonomy? So I wanted to quickly go over this as well. So I usually start with checking whether the phage that you have belongs to a new species. So our main species demarcation criterion is genome sequence identity of 95% threshold across the length of the genome, meaning that if two isolates differ from each other by less than 5%, then they belong to the same species. And one of the tools that is very easy to use for this is the tool called Viridic, which is the virus intergenomic distance calculator, where you can see the distance. For example, here everything was identical and it's 100, and it gives you 100%. And you can color this with whatever thresholds you like, but the basic settings are the ones for bacteriophages, being 95% for a species. It gives you a different color. So this top half just gives you the different intergenomic distances in percentages, and the left bottom left half gives you information about the aligned genome fraction and the genome length ratio. Let's say you've accidentally sequenced a phage and not accidentally sequenced a phage. Let's say you've accidentally assembled the phage genome twice, concatenated with each other. Then you can look here and you see, oh, this phage genome is much too long, and it's probably, you know, your aligned genome fraction and your genome length ratio will be wrong and will be flagged with some colors here. So then you have to be mindful for this, because you want to obviously have your just unique genome. Check for synteny. So if you're meaning the order of your genes, isolates with high level of rearrangements should not be in the same species. And if your phage is part of an existing species, it's very useful to have this information, because you can use it when you deposit your genome into GenBank or EMBL or DDBJ. So if your phage is the first isolate of a new species, then we can create that new species, but then we can also check whether your phage belongs to a new genus. So what is a genus? It's also just a group of viruses sharing a high degree of nucleotide sequence similarity, generally more than 70%, but it can also have additional defining characteristics, and we kind of look for verification by a phylogenetic tree of at least one of the signature genes. There's other elements that come into play here. You look at the average genome length, number of coding sequences, the genome organization, whether it has tRNAs, and then looking for certain signature genes and making a phylogenetic tree with appropriate in groups and out groups. If your phage belongs to is a first isolate of a new genus, you can contact me, or if it belongs to a specific a group of viruses for which we have study group chairs, you can contact your study group chair with the proposal, with help for creating a proposal. So then let's say your phage is really new and there's no species, there's no genus known. So then we go to, is it part of a new subfamily or a family? So subfamily is a level that is not mandatory to be created, but we find it useful in many cases where we see 2 or more genera recognized to be related, but below the level of the family. And usually it means that they still share some degree of nucleotide identity. Whereas for the family, it's really we're looking at shared proteins, amino acid identity, and so forth. So family is represented by a cohesive and a monophyletic group in any of our proteome-based clustering tools. It can be VIP3, Gravity, and so forth. Members of the same family share a significant number of orthologous genes, and we can't really put a number on it because there's a wide range of genome sizes, and a group of phages that are 15,000 base pairs or a group of phages that are 500,000 base pair might need some different numbers here. And an important thing to check is if your family level cluster shares orthologs with other family level clusters, you need to make sure that when you make a phylogenomic tree, so a tree, phylogenetic tree of the multiple genes, that tree makes your cluster robust and easily reproducible, high bootstrap values, and so on. So some older examples of when we did this and we published it are the family Cheetoviridae and the family Hirelviridae that you can look up in these publications. So one of the things that you can also use as a resource is the taxonomy browser at the ICTV. So it's at ictv.global in the taxonomy tab. And it has a browser like this where you can, for tailed phages, which you probably have, you can go and expand, look for the realm Duplo DNA viria, so Duplo 2 DNA, so it has double-stranded DNA. Go to the kingdom Hungongvirae, and then to the phylum Uroviricota, and this is where you find the class Cadoviricetes. So now if you start and look at, there's 1 order of bacteriophages and 3 orders of archaeal viruses, and then you can find all of the families that are not assigned to an order. So if I were to be interested in the family Acromanviridae, if you then hover over it, this is a screenshot from when I was hovering over this one with my mouse, you can see that it has 2 subfamilies, 10 genera, and 63 species. And if you then click for details, then you can actually see the taxonomy proposals related to this, and you can download it and look at, what the criteria were. So, for example, as I talked about Straboviridae and Cyanoviridae, you can have a look at it if you were to be so inclined. So what are the steps kind of that you want to go through in a flowchart? You want to start with a well-annotated phage. You want to find relatives in a database, and it could closest relatives. It can be relative, pun intended, being if you have a very, very novel phage, if you use any nucleotide search tools, like just blast in a database, you might not find any relatives, but if you look at the translated nucleotides, so the amino acid level or a protein level, then you might find some relatives. So it depends on which phage you have isolated, how new it is. So you want to look at first, if there are any relatives that have shared nucleotide identity, look at the intergenomic distances. Then you want to look at what protein content is shared. And then you want to do multiple sequence alignments of signature genes or multiple core genes. So for each of these steps, there are multiple tools to use, and really what I can suggest is do what you are comfortable with using. There's a lot of people who want to use web server. There's other people who use command line tools, they might not 100% match, but as long as you use the appropriate data and the appropriate outgroups with reproducible tools, you should be fine, whether you use something on a command line or on a web server. So if we look at how can that change in databases, I went to look back to one of the phages that I isolated for my PhD and deposited in the database in 2011. And at the time, this phage was classified as a virus, obviously. And then in the group double-stranded DNA viruses, no RNA stage, order Cadaveralis, family Podoviridae, subfamily Autographiviridae, and it was then grouped in the unclassified 5KMV-like phages. If we look at what it is now, and this is a screenshot that I actually took last year, we've seen that it's still in viruses, but these double-stranded DNA viruses are now in the realm Duplo-DNA-Viria. So then you have the whole lineage, Cadavericetes. So Cadaveralis is now gone. So I should have actually taken a new screenshot. And so Autographiviridae, family, and genus Limelight virus. So this is basic, but you can see that the date is still the same because it's the date from submission, and it's the NCBI that automatically does these updates with when there's a new taxonomy. Okay, so what's the process that we follow? So it's a long process because there's all of these checks and balances built in to make sure that the taxonomy that is created is high quality. So around May, there's a deadline for the submission of taxonomy proposals to your subcommittee chair, which in this case is me. Then there's a meeting of the executive committee who goes through all of the proposal of all of the subcommittees. So phages, archaeoviruses, eukaryotic viruses, single-stranded DNA, double-stranded DNA, RNA, all of these come together and we vote. Then there's time for corrections. Then we vote again by email for the corrections. And then there's a ratification vote by the entire ICTV membership, which usually happens January, February, or March. And then once that vote is done, there's a new taxonomy update. And this can be March or April, depending on this whole process, how long it takes and whether there's any issues identified. So at all stages, the proposals are available from our website. And they can be withdrawn by the authors if they realize they made a mistake, or they can be deferred to the next year if the corrections are too big to make in 1 year. But knowing this, that the best case scenario is that if you find a new phage and you propose, let's say, a new genus for it, then it's the best case scenario that it's formalized only 1 year after first reporting. So then I wanted to do the final couple of minutes on phage naming. So one of the big changes for all viruses is that we've changed to a binomial species naming format. And so remember, a species is a group of related phages that share more than 95% nucleotide identity across the genome. So it's basically a bin or a group of phages. It's in the sense that it's not the same as, you know, Homo sapiens as a species, where it's related to having fertile offspring, because it's very difficult to test this for our phages. So we basically have a genome-based species definition. So for that group, let's say Enterobacteria Phage T4, which is also known as Escherichia Phage T4 or Phage T4 or T4, is assigned to the species T. quattrovirus T4. So this binomial name consists of the genus name and then a species epithet. And that species epithet can be anything as long as it's one word and only uses Latin letters of the traditional Latin alphabet and numbers. So in this case, T. quattrovirus T4. Some taxonomy proposal authors choose to go for Latinized binomials, and this was the case for the Crassferale study group. So they had one of their phages was called, the phage name was uncultured crass phage because it was assembled as a genome from metagenomes, and it was assigned to the the species Cargivirus communis. And this is what it looks like then as how to write it. So if we look at, again, going back to my PhD, I isolated a phage that I called Dicea phage limestone 1, virus bacteria, Dicea solani, myovirus limestone 1. I commonly refer to it as limestone 1. You can find it here, and it's now assigned to the species limestone virus limestone, but there's loads of other phages assigned to the species as well. So depending on whether you work on a popular bacterium or a popular set of studies, this species might only have one member, or it might have loads of members. So you can see that the species limestone virus RC 2014 only has one member. So we try to write a paper that makes it clear, because it's now for all viruses, what the difference is between viruses and virus species by writing their names correctly. And basically, if we look at, for example, a maize mosaic virus in English, which infects maize, you see that your host species is Zea mays, and you use that in any language. But if you use your host name, it can be, you can change it in any other language. So this is basically the same for, this can be the same for your viruses or your bacteriophages. So this virus species is alpha-nucleorhabdis virus maydis. Regardless of where you are in the world, what language you speak, this should be the species name. But if you then look at the physical entity that causes the mosaic syndrome on maize, then it can be any, then this adapts according to your language. And I'm not gonna fall into the trap in trying to pronounce it here, but hopefully this illustrates the difference between your virus and your phage and your phage species. If we go back to, if you go back to here, you can see it here, the phage, Dicea phage, whatever you want to call it, CAMLD, associated with the species limestone virus limestone. So that's it. I hope I was clear enough, and sorry for the little bit of a mess up with the sharing the slides. I would like to thank my group here for being very supportive, and all my ICTV collaborators, and all of the funding bodies that fund my research. Thanks.
Thanks, Evelien. There are a lot of participants joining towards halfway or midway, but I would like to, maybe if you have read the paper before, maybe you have questions that you need clarity to, or so it's an open end, because a lot of people join towards the end. So I will start with the questions on the chat, and then I will go to the hand raisers.
Okay.
Yes, if that's okay. And there are quite a few hands being raised right now.
Okay, yeah, no,
okay.
Absolutely. Go ahead.
Yeah, okay. So how much percentage similarity determines a new family of a phage genome, and what tools can be used for a phage family?
Yeah. So we at the family level, we are looking at basically sharing a significant number of orthologous proteins. So proteins that are can be recognized by our current tools as being shared between all of the members of the family. Usually, if you take a family with very diverse membership, there's across the whole thing, there's no detectable nucleotide similarity anymore, but we can look at the amino acid similarities. So when I say a significant number, it's usually at least 10%. But we don't want to put an absolute number on it. As I said, it might be very different for genomes that are very small versus genomes that are very large. So the tools that we can use for this are any of the tools that look for shared proteins between two or a set of genomes, which can be, there's a lot of bacterial tools that are good that you can use. We've used in the past Pirate, Get Homologs, Rory, these types, any type of pan-genome tool if your set of phages has large genome, or the web server Core Genes has been very useful for us to identify shared proteins. And then once you have those, it's basically a matter of making multiple sequence alignments. So one of the tools that also kind of mimics this is VipTree. So VipTree doesn't look at the VipTree doesn't look at, I'm sorry, can you mute this person, Angela?
I'm trying to look for the person.
So where was I? Okay, so VipTree. VipTree does something similar, but it doesn't use the protein coding gene annotations. It just uses tBLASTX. So it uses the one genome, translates all 6 reading frames, and compares it to another genome, translates all 6 reading frames, and looks at it like that. But then it's much more difficult to put a percentage on that. I hope this answers the question.
I hope so too. Okay. Is there a criteria for naming the families of the phages?
No, there are no criteria. Well, yes and no. So we ask people to not name families or other taxa after themselves, but you can honor somebody else by naming it after them. But we ask that to be people who are not research active anymore, so retired or deceased, and the name can also not be offensive or a swear word or something like that.
And is it for phage people who've given their research into phage, or just anyone who's quite old in science or deceased?
So yeah, anybody, there needs to be a justification of why it is this name. So for example, we have a phage family called after Margarita Salas. We have one for Martha Chase. We have one for Giancarlo Scito. Often it's traced back to, one person who's isolated one phage a very long time ago, and this phage is now the typical phage, but it can be just, honoring somebody who's done something very interesting in science, or I try to stay away from any type of controversial people. I wouldn't suggest doing any kind of politicians or whatnot, but apart from that, we can have an open conversation about names.
All right. Of what importance is morph ID of viruses now?
So basically we think logging the morphology is still important, but we don't really know yet how it maps onto the full taxonomy, because we see that if you look at a very crude morphology like Podoviridae or Podovirus being a tailed phage with a short tail, that we saw is not representative, doesn't give good groupings, but it's possible that in future we could match this to these genome-based families if we go into much higher resolution of the morphology. If we, for example, can see that, okay, it has a base plate that's made up of this, and it has these type of tail fibers and tail spike, and if we like for a myofibrosis, and this type of color protein and stuff. So I think it's useful information to keep, but at the moment we're not mapping it onto the taxonomy.
All right, Camille Khan, if you can, he had several questions, so he asked if he could raise his hand. So
okay,
yeah.
Hello, madam. How are you? I hope you are fine and well.
Yes, thank you. Please go ahead with your question.
Madam, actually, I am from Pakistan and I have a lot of questions. But first of all, I have a question related to the phage therapy. Madam, the phage taxonomy, whatever you explained, is there any advantages of the phage taxonomy on the phage therapy?
Yeah, I think it's a good question. So what we've realized is that when we have a good taxonomy where all the groups of phages are clearly separated based on their genome, that we are reiterating kind of phenotypic differences as well. So we can use this, for example, when we find that this family of phages is very efficient at generalized transduction. So we might want to say phages belonging to this family we shouldn't use for phage therapy, for example. So we will find this out as the taxonomy progresses and as we have more information about successful and unsuccessful phage therapy trials. And hopefully we can use this in the future to kind of make predictions of what a good phage or what a bad phage might look like for therapy.
Madam, I have another question from.
Yes.
Madam, my second question is that the phages means the phage which you isolate. Did you apply it on the did you apply it on phage therapy? Means did you use it in the phage therapy, or no? You just explain, or you just publish the paper on phage taxonomy. I mean that you used your phages on phage therapy, or no?
So this what I presented now is a community effort of dozens of people across the world based on the genomes. So this is done with publicly available phage data. So this is not in a phage therapy context.
Madam, another question is that, Madam, actually I am a student, so that's why I have a lot of questions. I hope you will not mind it. My third question is that, Madam, is there any phages that we have only single-stranded genome, like a single-stranded DNA or single-stranded RNA? Actually, I have listened that there is a phages they have double-stranded genome, like double-stranded DNA, double-stranded RNA. Or no, we have also phages that they have also single-stranded and single-stranded genome as well. If they have, if we have, so give me a little bit example or the name of the phages.
It feels like you're asking me to do your literature research for you, but I'll briefly answer. So what I talked about now was specifically about double-stranded DNA bacteriophages with head-tailed morphologies, which is about if you isolate a new phage, it's likely going to look like this. Phages come in a lot of different genome organizations and morphologies. So there is positive sense single-stranded RNA phages, class levivirucides. There is single-stranded DNA bacteriophages. The most famous one is probably Phage 174. There are double-stranded RNA phages. There are filamentous single-stranded DNA phages. There's many different ones.
Thank you, madam. Thank you so much. I got a lot of things and I get everything, sir. And I really appreciate your effort. And mashallah, you explain each and everything very well. Actually, I'm a student. That's why I don't know about each and everything. But the presentation, what you present here, I get a lot of knowledge. Thank you, madam.
My pleasure.
Nkechi, do you want to proceed with the questions? Nkechi? All right. If she's not available, so does the updated classification put proteomic findings into use to distinguish between the phages?
Yeah, absolutely. We use.
Hello?
Oh, sorry.
Okay. Nkechi, you'll continue on with the question. Just ask one more question, and then Evelien will answer.
I have a question, but Camille has already asked the question.
Okay.
It's about of what importance is phage taxonomy to phage therapy. You've already answered the question, so
Okay.
I'm okay by that.
Okay. So I'll answer the question on the proteomic findings. So basically, we use predicted proteome findings currently in our taxonomy, meaning we use the genome and predict the proteins and look at it like that. If we were to have proper proteomic, experimental proteomic data, that would be even, that adds another layer of information, and we can use that to annotate further and refine further the taxonomy. But so we don't have an approach to do it right now directly from proteomic data, but we use predicted proteomic data, if that makes sense to you.
All right. Another question is, how does one construct how does one choose proteins to construct proteomic phylogenetic tree for phage classification? Does it need to be one or more trees with different proteins?
Excellent question. So there's different approaches to this. If you use only one protein, then you have then there's the potential that this protein does not represent the whole genome, because it might have been more subject to horizontal gene transfer than, for example, other proteins. So it's always good practice to use more than one protein. When in doubt, terminase large subunit, portal protein, capsid protein are good proteins to use. If you take a more systematic approach, then you can take your whole group of viruses that are phages that you look at, predict all of the proteins, and then compare them all against all and see which proteins they share. And that case, so that gives you the core proteins, and then you can either select from those core proteins or use all of them to make your multiple sequence alignments and then your trees.
All right. I do hope we have answered questions. The next time, Evelien will give you some e-water. Yeah, and I hope everyone's questions have been answered, and it was a really lovely presentation. And I think there's a comment where we need.
Question.
Yes, Emmanuel, go ahead.
Yeah, so I thank you very much, Evelien, for that presentation. I'm asking, I'm trying to find out what is wrong with using whole genome sequences for phylogenetic tree analysis?
Oh, there's nothing wrong with using whole genomes, but phages are very diverse in their genomes. So if you use whole genomes and there's no more nucleotide similarity, similarity at the nucleotide level between your furthest apart genomes, then your clustering will never be as good as if you use the amino acid level where the diversity would be lower. So, for example, if you look at only within a species and you want to do, a fine grain analysis of the whole genome and put that into a tree, like if you were to make a SNP tree for bacteria, then absolutely you can use the whole genome. It's just to we move on to the kind of the protein and amino acid level because the diversity is so large.
But can you use a SNP tree for phages as well? Because I'm trying to remove the issue of the challenges of getting the core protein and all of that. Can the SNP tree also work well?
I guess a SNP tree would really only work if you have, let's say, 100 or like more than 10 genomes that are very, very similar. So I've never really encountered a situation where that's the type of analysis that I can do. It's more like everything's too diverse. So we go to the protein level. So it depends on your data set. If you, for example, do some sort of evolution experiment with a set of really related phages, then I can totally see a SNP tree being useful.
And now you have an e-coffee.
Yes, I can see it in the chat. Well, I can still drink some of my water with phages on it.
Yes. And does anyone have any more questions? Because we are close to the hour.
Hello.
Yes,
Hello.
hi Newton. Yes.
Yes, thank you, Angela, for moderating this meeting, and thanks, Evelien, for your time. I'm always happy to have you on board. I think we have some people who made our time to really attend this meeting. We are having Dr. Rami Aziz there, and great to see you, Evelien. And you know, yes, and yes, he's there.
Hi, Rami.
Yes, they are showing people on the forum. We recognize the president of Jessica Nakaguma as well, and Lillian. They are there. So thanks so much for attending this meeting. And hand over to Angela to continue. Thanks.
Yeah, thank you, Evelien. Thank you, Remy, and thank you, Jessica. But Evelien, just a quick question in terms of capacity, because now you have a few local chairs. Local chairs would mean the executive committee, and then now you have, I think, Jessica and Remy from Africa. How do you think we would expand on this?
So the way the subcommittee is currently set up is, so there's a vice chair, there's a chair which is me, vice chair which is Dan Turner. Then we have a number of study group chairs who are responsible for a subsection of, related phages. For example, we have a Heralverde study group chair, we have a T4-like study group chair, these type of things. And then we have a number of regional representatives, and we try to get as much spread across the world as possible. So Rami and Jessica are regional representatives. I do hope that in the future we can get more study group chairs from different places in the world. So anybody who is interested in doing this can contact me. We currently work with kind of 3-year terms. So coming at the end of 2023, we will renew. And I'll probably be looking for new people to join. So if anybody's interested, then you can just send an email to me.
All right. I see Rami's hand is up. Hi,
No,
Rami.
I was just saying that just maybe this week, or I don't know, maybe a couple of weeks ago, Andrew Krupinski contacted us to that they are going to name a new genus over some institute in Egypt. So there was a bit of debate. They wanted to name it on the institute name, but the name belongs to a person who is a Nobel laureate. So actually they didn't like to have his name connected to a virus. Yeah, as if virus is a negative thing. So it ended up being called Giza virus, I think. So that will be a name of a genus. Giza is the area where the pyramids are. So that's where the institution is also. So I think that's one difference from 10 years ago, I think. So now you will have more and more names from other continents. That's an example of how the outreach is working.
Yeah, exactly.
Camille, I don't know whether this is an old hand or a new hand.
Madam, I have some question. Actually, Madam, if you don't mind, can I actually need a contact number because I want to contact you for PhD microbiology if there is any post for biology.
All right.
Already my Master of Philosophy from Hazara University, and I already done my phage therapy, and alhamdulillah, I already published almost 3 papers. So I actually really interested in your presentation. So that's why I need a little bit contact with you if you contact with me.
Yeah, so personally, I only accept PhD students if I have funding, and then that funding is announced on our website. So so that's for everybody who's interested, and I know at least one person in the room who has been successful using that route. So that's that, but I will give my email address out to everybody, but please, I cannot help everybody with every little thing. You need to contact your own supervisors and your own, university departments first before you contact me. I'm gonna type it in the chat now.
Madam, may I ask another question? Actually, email is too much busy things, and when we email to someone, so they reply very late, or sometimes they will not reply. But WhatsApp and other like Skype,
I will not give my WhatsApp. I will not give my phone number. This will not happen.
Right.
But so
Evelien has shared her email on the chat. So if you need advice or anything in terms of proteomics, phage annotation, or if you have sequencing and you're wondering what type of sequencing you would like to do, I think she'll be the most appropriate person.
I mean, I would encourage everybody to also kind of read the papers and all of this, because a lot of information can already be found in there. If it's difficult, please send me an email. I do get loads of emails, so I might be delayed in responding. But
excuse Evelien, I have one last question.
Yes, Nitin.
Excuse me. If someone have published on Phage taxonomy, basically publish and classify phages as maybe podovirian, and then you have now abolished this family, and the person is actually carrying out phage therapy. Will he be in position really to use that previous finding?
So what really happens is you have your phage, it has its genome sequence and it has its name. That will never change, right? It's just the kind of bin that we put it in will change. So it should not have direct consequences for any phage therapy, because your phage name and your genome sequence will remain as it was published. So it could be updated in any type of protocols later on. But this is why there should be a difference between the phage species and all of the other taxa and the phage name and the sequence. So. But now I'm gonna have to go.
Yes, thank you, Evelien, for everything, and would encourage everyone to just probably we'll have as APF, we're going to try to have a small training on, and we'll invite people to it's probably going to be a webinars. So we're thinking about that. So we just have to get our directions right and to encourage people to attend so that you may be able to understand a lot of things on proteomics and how to name your phages. And probably Evelien will again have to give this talk at some later points in the year. Evelien, thanks so much.
It was my pleasure, Angela.
Yes, and everybody's saying thank you, and it's such a wonderful presentation, and we hope we'll have you soon.
All right.
In terms of training for proteomics and just alignment and everything.
Okay, excellent. We'll see what we can do.
All right.
And great to see you all, and have a nice day, evening.
You too.
Thank you.
Bye-bye.
Very much.
Bye.